Hello, everybody, and welcome back. Today we're going to start our discussion on DNA and its role in heredity and the inheritance of traits. As we talked about in our last unit of genetics, we all know that traits are passed from one generation to the next, and that's what Gregor Mendel studied with his pea plants and other geneticists followed up with over the years. But one piece of information that Gregor Mendel didn't have, which actually makes it really impressive, the discoveries he made, even at the end of his days, he didn't know what it was that was actually passing that genetic information along. He just knew that there was something being passed along. And this shows the big difference between what we now know about DNA and what was known just a few decades ago. Really, all of this that we're going to be talking about today wasn't discovered until about 60 years ago, which in the span of time that humans have been experimenting with science, 60 years is not that much time. So the things that you're going to be learning about and some of the things you already take for granted are things that university level researchers were just discovering in the 50s and 60s. So what you're looking at here, this is obviously a model of DNA. You've all seen pictures that look something like this. And there's a couple different terms that are used to describe DNA. One of them is a twisted ladder. So if you can imagine, this blue part here is kind of like the sides of a ladder. And of course, there are two sides to a ladder. And then the parts in the middle, these would be like the rungs of the ladder. So these are the parts that you hold on to. And if you kind of imagine it, you can see how this looks like a ladder that's been sort of twisted up on itself. And then the other name that's used for DNA is probably one you've heard before, which is a double helix. Double referring to the fact that there's two sides to the molecule, and helix referring to that kind of twisted structure that you're looking at here. So again, it wasn't until kind of the early 1920s that the first leaps and bounds were being made towards discovering that DNA was in fact the molecule of heredity. For a long time, it was actually believed that proteins were the molecule of inheritance, mainly because of the complexity. Most scientists looked at proteins and saw there's 20 different amino acids. They can be arranged in millions of different combinations. And here's DNA, this molecule, it's only made of 4 monomers. And how could only 4 building blocks make something as complex as a human being? Well, we now know that that is actually the case. And one of the first big steps along the way was using very simple techniques like staining cells and looking at them under a microscope. And so scientists in the early 20s found that there was a particular stain that would stain the DNA this reddish color. And the more intense that stained color was, the more DNA was in that location. So they could use it to identify the location and relative amounts of DNA. And that led them to 3 really important pieces of evidence for genes being found in the DNA. The first piece of evidence was that the DNA was always in the right place. They knew the nucleus was really important for heredity and inheritance of genes, and the DNA was always found in the nucleus. It was also found in the right amounts. And so if they compared somatic cells of organisms to the gamete cells of those same organisms, they found that there was roughly double the amount of DNA in the somatic cells as compared to the gametes. And this really fit that idea that gametes were haploid, and then they fused together during fertilization to create diploid cells. And then finally, when comparing various species, they noticed that they had varying amounts of DNA. So anywhere from bacteria, whose DNA is only about 1 millimeter in length, if you were to stretch it out, all the way up to, for example, humans, where our DNA is about 6 feet long if you stretched it all out. But different species have different amounts of DNA. So this was just kind of the launching point. And then from there, there was a series of experiments to show that DNA was in fact the molecule of inheritance. And that's mainly what we're going to talk about today, are these experiments that showed DNA to be the molecule of inheritance. So the first of these experiments was done by a scientist named Frederick Griffith. And his experiments took place way back in 1928. And so what he did was he was studying Streptococcus bacteria, so the same bacteria that causes strep throat. And so what he did was he isolated 2 different strains of strep bacteria. There was one that he called the S strain, and one was the R. The S strain is the wild type. This is the one that's virulent. So this is the one that can make you sick. This is the one that can kill you. The R strain lacked some enzymes that gave it that really thick cell wall that would protect it from antibiotics and from our immune system. And so this R strain, if organisms were infected with it, it was basically harmless. And so it didn't really do anything to individuals who were tested. And so the first thing he did was he took some of that S strain bacteria, he injected it into mice, and the mice died. So this experiment taught us a couple things. It taught us, number one, it's really bad to be a mouse in a laboratory. And number two, it really highlighted some important things about DNA. And so when he took the R strain and injected that into mice, they lived. The S and the R just refer to the fact that the S strain, when he grows them in a petri dish, would form smooth-edged colonies, whereas the R strain would form rough-edged colonies, and that's where we got that designation. So this is where the experiment really got interesting. He took those S strain, the bacteria that caused the infection and caused the death of the mice, and he boiled them. And he found that when he boiled these bacteria, it basically killed those bacteria. And when he injected them into the mice lived. It was totally harmless to them. And even more so, he found no evidence of those bacterial cells in their blood. With this first group, after the mice had died and they autopsied them, they found that these bacteria were proliferating in their cells. Here, there was no bacteria found, and here, there was also no bacteria found. And then, being a good scientist, he decided to take this test one step further, and this is where the real discovery was made. So he took these heat-killed bacteria, and he took the R strain bacteria, both of which by themselves caused no harm to the mice, and then he mixed them together into a solution. And what he found then was that the mice died when they were exposed to them, and there was bacteria. The live S strain bacteria was found inside the tissues of these mice. So somehow these heat-killed bacteria had transformed these harmless bacteria into virulent bacteria. So his conclusion that he came up here with was that a chemical substance from one cell can genetically transform another cell. And that's exactly what we call this process when the DNA of one molecule changes another, or DNA of one organism changes another, we call that process transformation. And so several years later, in the 1940s, 1944 to be exact, another group of scientists led by a scientist named Oswald Avery decided that they wanted to take this experiment that Griffith did and kind of take it a little bit further and confirm the results, because people still didn't believe that it could be DNA that led to inheritance. So what they did was, again, he took some of that strep bacteria, he boiled it, but then he just isolated the liquid. He got rid of all the cellular debris, and he just isolated what was left behind in that liquid. So when he boiled the bacteria, they would break apart, and he would strain out all the membrane components and all the other stuff that was there and just leave behind this liquid. And then he treated that liquid with 3 different enzymes. He treated them with RNase enzymes, which break down RNA. He treated them with protease enzymes, which break down protein. And then he treated them with DNase enzymes, which break down DNA. In the group that was treated with RNA degrading enzymes, the heat-killed bacteria, the liquid that was isolated, was mixed with that R strain bacteria, and he ended up with virulent R strain bacteria, or virulent S strain bacteria. With the protease group, he found the same thing. He mixed them with the harmless R strain bacteria, and he ended up with virulent S strain bacteria at the end. It wasn't until he treated them with DNA degrading enzymes that there was no change and that the R bacteria remained those harmless R strain bacteria. So this experiment kind of gave results on 2 fronts. Number one, it eliminated other possible molecules of transformation. RNA, being a nucleic acid, was also believed to be one of the molecules of inheritance, and this experiment showed definitively that that was not the case. As we talked about earlier, protein was also thought to be a molecule of inheritance, and this showed that it was not the case. So by the process of elimination, it showed us that DNA must be the molecule. And this experiment also did a good job of providing positive evidence of that. And that happened up here where we strained out that liquid. So all that other cellular debris was eliminated, and really all that was left in this flask was some fluid with DNA in it. And so his final conclusion that he came up with here was that since DNA destroyed the transforming substance, that transforming substance must be DNA. And then finally, about 8 years later, in the year of 1952, and I don't expect you to memorize these dates, by the way. In 1952, scientists by the name of Hershey and Chase conducted one more experiment to kind of put the last little pieces together here. And in their experiment, they used something called a bacteriophage, which is a virus that infects only bacteria. And these viruses are very, very simple in structure. All they have is a protein coat and some genetic material, and that's it. They're just DNA and protein, nothing else. They rely on the bacterial cells to actually provide the rest of what they need to make more bacteria, or viruses, I should say. So Hershey and Chase did a very ingenious experiment here to isolate whether it was protein or DNA that was causing the transformation. They grew 2 batches of viruses. And this first batch of viruses was grown with a radioactively labeled phosphorus. And we know that DNA has a lot of phosphorus in it because of its phosphate groups. So they were essentially radio labeling that DNA. The next group was grown with radioactively labeled sulfur. And we know that some proteins have sulfur in them. So this was basically radioactively labeling this protein coat of our viruses. And so after exposing these viruses to the bacteria, they were put into a blender. And basically what the blender did was just strip away any of the viral components that were left on the surface of the bacteria and just left those bacterial cells intact. And then after spinning those through the centrifuge, they found that down here at the bottom, these little pellets were the bacteria themselves. And in the group that had the radioactively labeled phosphorus, all of that phosphorus was found down in the bacteria. So what that told them was that it was the DNA that was in the viruses that actually infected the bacteria and not the proteins. So after all of these experiments, the structure of DNA really only needed a couple more pieces of the puzzle to figure out how it was laid out. And one of the last pieces of information that really tied this all together was discovered by this scientist by the name of Rosalind Franklin. And she studied something called X-ray diffraction, where samples of DNA are crystallized, they shine X-rays through those crystals, and then the diffraction patterns, which is what you see right here, illustrate some of the key features of that DNA structure. And so what her X-ray diffraction photos showed by these bands and repeating patterns was that the DNA had this helical pattern and it had a very specific diameter as well. So she was able to see some really specific pieces of information about that DNA strand. Another scientist working around the same time was named Erwin Chargaff, and he and his contemporaries were studying samples of DNA from as many different sources as they could get. And they always found that the amount of adenine in DNA matched up with the amount of thymine, and the amount of guanine always matched up with the amount of cytosine. And they put the pieces together that adenine and guanine fell into this group called purines, and thymine and cytosine fell into this group called pyrimidines. And furthermore, they found that purines always bonded with pyrimidines, which accounted for the fact that they occurred in equal amounts. And this was found so extensively that they actually coined the term of Chargaff's rule to say that A is always present in equal amounts as T, and G is present in equal amounts as C. Another thing you'll notice here is that these purines actually have a double ring structure, whereas the pyrimidines just have a single ring structure on their nitrogenous base. And we'll get into that a little bit more, why that's important later on. And finally, Watson and Crick kind of took all of this information and put it together to build the first three-dimensional model. And that's actually what you see over here is them standing there with the actual model that they came up with. So James Watson and Francis Crick were the two scientists who were credited with the discovery of DNA, but in reality, it relied on the discoveries of a lot of other scientists as well. And so there was a lot of other folks who really put in some input in order to discover the structure of DNA. And again, so Chargaff's rule was something that really helped them fit together the final little pieces of this structure because it established what we now know as the rules of base pairing, which again says that adenine always goes with thymine and guanine always goes with cytosine and vice versa. And it's not the other way around. That X-ray diffraction that Rosalind Franklin did really helped with that discovery because, as I said, she realized there was a set diameter of DNA. And if there was ever a time where adenine and guanine bonded together and cytosine and thymine bonded together, you would see this kind of change in the diameter of the molecule. And that was never observed through any of the tests that were done. And so from their studies, they found some key structural features of the DNA. The first of those was that DNA is a double-stranded helix. And so we've kind of talked about that already. You can see here, here's one backbone of one strand kind of bending that way, and here's the other one making this sort of pattern with the nitrogenous bases connecting them in the middle. One of the other key features is that it's a right-handed helix. So if you were to use your right hand as a guide and stick your thumb up in the air, as the strand goes up, that DNA strand would coil around that way. There's not really a whole lot of significance to that for our purposes, but it is one of the key features that was discovered. The main reason for that is the hydrogen bonding. And you'll notice that between adenine and thymine, they always form 2 hydrogen bonds, whereas cytosine and guanine always form 3. And so kind of that uneven bonding leads to that twisting shape of the molecule. Along with that, there's van der Waals forces. So the hydrogen bonds are holding together the bonds this way between the rungs of the ladder, whereas van der Waals forces kind of attract between adjacent nucleotides this way. And so all of those bonds together hold that molecule, hold that shape together really tightly. And the last key feature that we're going to talk about is the fact that DNA is anti-parallel. We're going to get into this a lot more when we get to how DNA replicates itself. But for right now, it's just good to understand that in DNA, it has a sugar called deoxyribose. That's where the deoxyribo in the deoxyribonucleic acid comes from. And that's what we're looking at here. And you'll notice the carbons are numbered, 1, 2, 3, 4, and 5 prime. And so the phosphate group of one nucleotide is going to bond to this 5 prime carbon. And then the next sugar in the line is going to bond to that 3 prime carbon. And so what ends up happening is on one side of the strand, you'll have a 5 prime carbon hanging off on one end and a 3 prime on the other. And the other side of the DNA molecule, those sugars are basically upside down in their orientation. So they're pointing the other way. So on this side where you have 5 prime on the top and 3 prime on the bottom, on this side you have 3 prime on the top and 5 prime on the bottom. And when we get to DNA replication, again, we'll talk about why that's really significant. And the last thing I want to talk about today is just why the shape of the molecule is really essential to the function of the molecule. And that boils down to 4 main things. Number one, and again, this is something we kind of take for granted now, but DNA stores genetic information. DNA in humans is roughly about 4.5 billion base pairs, which means there's a ton of information that can be stored in that DNA molecule. Different species have different amounts, but humans' DNA is about 4.5 billion base pairs. Just to put that into perspective, if you were to read each letter of your DNA, 1 letter per second, it would take you over 90 years to read your strand of DNA. So it is a huge molecule. DNA is also susceptible to mutations. And mutations can lead to genetic diversity, which when we get to ecology and evolution, we'll talk about why this is so hugely important. So the fact that mutations happen is actually a beneficial thing in terms of evolution. DNA is also precisely replicated in cell cycles. So again, imagine 4.5 billion base pairs. If you had to copy that by hand, it would take you years to do each strand. And your cells can do it basically in a matter of hours, which is amazing. And the last thing, DNA is expressed as the phenotype. Again, phenotype is your physical characteristics. And the structure of DNA doesn't really show you how this happens. But when we get to protein synthesis in the later part of this unit, that'll make this a little bit more clear. But for now, that's everything about DNA structure. Our next video will go into how DNA copies itself, but that's going to be it for now.